Superphylum Ecdysozoa. Superphylum Ecdysozoa. The Superphylum Ecdysozoa contains an incredibly large number of species. This is because it contains two of the most diverse animal groups. Phylum Nematoda, the roundworms, and Phylum Arthropoda, the arthropods. The most prominent distinguishing feature of ecdysozoans is their tough external covering called the cuticle. The cuticle provides a tough but flexible exoskeleton that protects these animals from water loss, predators and other aspects of the external environment. All members of this superphylum periodically molt, or shed their cuticle as they grow. After molting, they secrete a new cuticle that will last until their next growth phase. The process of molting and replacing the cuticle is called ecdysis, which is how the superphylum derived its name. Phylum Nematoda. The Nematoda, like most other animal phyla, are triploblastic and possess an embryonic mesoderm that is sandwiched between the ectoderm and endoderm. They are also bilaterally symmetrical, meaning that a longitudinal section will divide them into right and left sides that are symmetrical. Furthermore, the nematodes, or roundworms, possess a pseudocoelom and consist of both free-living and parasitic forms. It has been said that were all the non-nematode matter of the biosphere removed, there would remain a shadow of the former world in the form of nematodes, the arthropods. One of the most successful taxonomic groups on the planet are coelomate organisms characterized by a hard exoskeleton and jointed appendages. Both the nematodes and arthropods belong to the superphylum Ecdysozoa that is believed to be a clade consisting of all evolutionary descendants from one common ancestor. The name derives from the word ecdysis, which refers to the shedding, or molting, of the exoskeleton. The phyla in this group have a hard cuticle that covers their bodies, which must be periodically shed and replaced for them to increase in size. Phylum Nematoda includes more than 28,000 species, with an estimated 16,000 being parasitic in nature. The name Nematoda is derived from the Greek word nemos, which means thread, and includes roundworms. Nematodes are present in all habitats, with a large number of individuals of each species present in each. The free-living nematode, Caenorhabditis elegans, has been extensively used as a model system in laboratories all over the world. Morphology. In contrast with cnidarians, nematodes show a tubular morphology and circular cross-section. These animals are pseudocoelomates and show the presence of a complete digestive system with a distinct mouth and anus. This is in contrast with the cnidarians, where only one opening is present, an incomplete digestive system. The cuticle of nematodes is rich in collagen and a carbohydrate protein polymer called chitin, and forms an external skeleton outside the epidermis. The cuticle also lines many of the organs internally, including the pharynx and rectum. The epidermis can be either a single layer of cells or a syncytium, which is a multinucleated cell formed from the fusion of unenucleated cells. The overall morphology of these worms is cylindrical, as seen in Figure 1. The head is radially symmetrical. A mouth opening is present at the anterior end with 3 or 6 lips as well as teeth in some species in the form of cuticle extensions. Some nematodes may present other external modifications like rings, head shields, or warts. Rings, however, do not reflect true internal body segmentation. The mouth leads to a muscular pharynx and intestine, which leads to a rectum and anal opening at the posterior end. The muscles of nematodes differ from those of most animals. They have a longitudinal layer only, which accounts for the whip-like motion of their movement. Excretory system. In nematodes, specialized excretory systems are not well developed. Nitrogenous wastes may be lost by diffusion through the entire body or into the pseudocoelom body cavity, where they are removed by specialized cells. Regulation of water and salt content of the body is achieved by rennet glands. Present under the pharynx in marine nematodes. Nervous system. Most nematodes possess 4 longitudinal nerve cords that run along the length of the body in dorsal, ventral, and lateral positions. The ventral nerve cord is better developed than the dorsal or lateral cords. All nerve cords fuse at the anterior end, around the pharynx, to form head ganglia or the brawn of the worm, which take the form of a ring around the pharynx, as well as at the posterior end to form the tail ganglia. In C. elegans, the nervous system accounts for nearly 1/3 of the total number of cells in the animal. Reproduction. Nematodes employ a variety of reproductive strategies that range from monoecious to dioecious to parthenogenic, depending upon the species under consideration. C. elegans is a monoecious species and shows development of ova contained in a uterus as well as sperm contained in the spermatheca. The uterus has an external opening known as the vulva. The female genital pore is near the middle of the body, whereas the male's is at the tip. Specialized structures at the tail of the male keep him in place while he deposits sperm with copulatory spicules. Fertilization is internal. An embryonic development starts very soon after fertilization. The embryo is released from the vulva during the gastrulation stage. The embryonic development stage lasts for 14 hours. Development then continues through 4 successive larval stages with ecdysis between each stage L1, L2, L3, and L4, ultimately leading to the development of a young male or female adult worm. Adverse environmental conditions like overcrowding and lack of food can result in the formation of an intermediate larval stage known as the dower larva. Everyday connection. C. elegans, the model system for linking developmental studies with genetics. If biologists wanted to research how nicotine dependence develops in the body, how lipids are regulated, or observe the attractant or repellent properties of certain odors, they would clearly need to design 3 very different experiments. However, they might only need one object of study. C. elegans. The nematode, Caenorhabditis elegans, was brought into the focus of mainstream biological research by Dr. Sidney Brenner. Since 1963, Dr. Brenner and scientists worldwide have used this animal as a model system to study various physiological and developmental mechanisms. C. elegans is a free-living organism found in soil. It is easy to culture this organism on agar plates. 10,000 worms plate. It feeds on Escherichia coli, another long-term resident of biological laboratories worldwide, and therefore, it can be readily grown and maintained in a laboratory. The biggest asset of this nematode is its transparency, which helps researchers to observe and monitor changes within the animal with ease. It is also a simple organism with fewer than 1,000 cells and a genome of 20,000 genes. It shows chromosomal organization of DNA into 5 pairs of autosomes plus a pair of sex chromosomes, making it an ideal candidate to study genetics. Since every cell can be visualized and identified, this organism is useful for studying cellular phenomena like cell-cell interactions, cell fate determinations, cell division, apoptosis, and intracellular transport. Another tremendous asset is the short life cycle of this worm. Figure 2. It takes only 3 days to achieve the egg to adult to daughter egg. Therefore, tracking genetic changes is easier in this animal. The total lifespan of C. elegans is 2 to 3 weeks. Hence, age-related phenomena are easy to observe. Another feature that makes C. elegans an excellent experimental model system is that the position and number of the 959 cells present in adult hermaphrodites of this organism is constant. This feature is extremely significant when studying cell differentiation, cell-cell communication, and apoptosis. Lastly, C. elegans is also amenable to genetic manipulations using molecular methods, rounding off its usefulness as a model system. Biologists worldwide have created information banks and groups dedicated to research using C. elegans. Their findings have led, for example, to better understandings of cell communication during development, neuronal signaling, and insight into lipid regulation, which is important in addressing health issues like the development of obesity and diabetes. In recent years, studies have enlightened the medical community with a better understanding of polycystic kidney disease. This simple organism has led biologists to complex and significant findings, growing the field of science in ways that touch the everyday world. A number of common parasitic nematodes serve as prime examples of parasitism. These animals exhibit complex life cycles that involve multiple hosts, and they can have significant medical and veterinary impacts. Humans may become infected by Dracunculus metanensis, known as guinea worms, when they drink unfiltered water containing cope pods. Figure 3. Hookworms, such as Ancylostoma and Necator, infest the intestines and feed on the blood of mammals, especially in dogs, cats, and humans. Trichina worms, Trichinella, are the causal organism of trichinosis in humans, often resulting from the consumption of undercooked pork. Trichinella can infect other mammalian hosts as well. Ascaris, a large intestinal roundworm, steals nutrition from its human host and may create physical blockage of the intestines. The filarial worms, such as Dirofilaria and Wuchereria, are commonly vectored by mosquitoes, which pass the infective agents among mammals through their blood-sucking activity. Dirofilaria amidis, a blood-infective parasite, is the notorious dog heartworm species. Wuchereria bancrofti infects the lymph nodes of humans, resulting in the non-lethal but deforming condition called elephantiasis, in which parts of the body become swelled to gigantic proportions due to obstruction of lymphatic drainage and inflammation of lymphatic tissues. Phylum Arthropoda. The name Arthropoda means jointed legs. In the Greek, arthros means joint, and podos means leg. It aptly describes the enormous number of invertebrates included in this phylum. Arthropoda dominate the animal kingdom with an estimated 85% of known species included in this phylum and many arthropods yet undocumented. The principal characteristics of all the animals in this phylum are functional segmentation of the body and presence of jointed appendages. Arthropods also show the presence of an exoskeleton made principally of chitin, which is a waterproof, tough polysaccharide. Phylum Arthropoda is the largest phylum in the animal world, and insects form the single largest class within this phylum. Arthropods are eucoelomate, protostomic organisms. Phylum Arthropoda includes animals that have been successful in colonizing terrestrial, aquatic, and aerial habitats. This phylum is further classified into 5 subphyla: Trilobitomorpha, trilobites, all extinct; Hexapoda, insects and relatives; Myriapoda, millipedes, centipedes, and relatives; crustaceans, crabs, lobsters, crayfish, isopods, barnacles, and some zooplankton; and Chelicerata, horseshoe crabs, arachnids, scorpions, and daddy longlegs. Trilobites are an extinct group of arthropods found chiefly in the Precambrian era that are probably most closely related to the Chelicerata. These are identified based on fossil records. Figure 4. Morphology. A unique feature of animals in the arthropod phylum is the presence of a segmented body and fusion of sets of segments that give rise to functional body regions called tagma. Tagma may be in the form of a head, thorax, and abdomen, or a cephalothorax and abdomen, or a head and trunk. A central cavity, called the hemocoel, or blood cavity, is present, and the open circulatory system is regulated by a tubular or single-chambered heart. Respiratory systems vary depending on the group of arthropod. Insects and myriapods use a series of tubes, tracheae, that branch through the body, open to the outside through openings called spiracles, and perform gas exchange directly between the cells and air in the tracheae. Whereas aquatic crustaceans utilize gills. Terrestrial chelicerates employ book lungs, and aquatic chelicerates use book gills. Figure 5. The book lungs of arachnids, scorpions, spiders, ticks and mites, contain a vertical stack of hemocoel wall tissue that somewhat resembles the pages of a book. Between each of the pages of tissue is an airspace. This allows both sides of the tissue to be in contact with the air at all times, greatly increasing the efficiency of gas exchange. The gills of crustaceans are filamentous structures that exchange gases with the surrounding water. Groups of arthropods also differ in the organs used for excretion, with crustaceans possessing green glands and insects using Malpighian tubules, which work in conjunction with the hindgut to reabsorb water while ridding the body of nitrogenous waste. The cuticle is the covering of an arthropod. It is made up of two layers. The epicuticle, which is a thin, waxy water-resistant outer layer containing no chitin, and the layer beneath it, the chitinous procuticle. Chitin is a tough, flexible polysaccharide. In order to grow, the arthropod must shed the exoskeleton during a process called ecdysis. To strip off. This is a cumbersome method of growth, and during this time, the animal is vulnerable to predation. The characteristic morphology of representative animals from each subphylum is described below. Subphylum Hexapoda. The name Hexapoda denotes the presence of 6 legs, 3 pairs, in these animals as differentiated from the number of pairs present in other arthropods. Hexapods are characterized by the presence of a head, thorax, and abdomen, constituting 3 tagma. The thorax bears the wings as well as 6 legs in 3 pairs. Many of the common insects we encounter on a daily basis, including ants, cockroaches, butterflies, and flies, are examples of hexapoda. Amongst the hexapods, the insects, figure 6, are the largest class in terms of species diversity as well as biomass in terrestrial habitats. Typically, the head bears 1 pair of sensory antennae, mandibles as mouthparts, a pair of compound eyes, and some ocelli, simple eyes, along with numerous sensory hairs. The thorax bears 3 pairs of legs, 1 pair per segment, and 2 pairs of wings, with 1 pair each on the second and third thoracic segments. The abdomen usually has 11 segments and bears reproductive apertures. Hexapoda includes insects that are winged, like fruit flies, and wingless, like fleas. Subphylum Myriapoda. Subphylum Myriapoda includes arthropods with numerous legs, although the name is hyperbolic in suggesting that myriad legs are present in these invertebrates. The number of legs may vary from 10 to 750. This subphylum includes 13,000 species. The most commonly found examples are millipedes and centipedes. All myriapods are terrestrial animals and prefer a humid environment. Myriapods are typically found in moist soils, decaying biological material, and leaf litter. Subphylum Myriapoda is divided into 4 classes: Chilopoda, Symphyla, Diplopoda, and Pauropoda. Centipedes like Scutagera coleoptrata, figure 7, are classified as chilopods. These animals bear 1 pair of legs per segment, mandibles as mouthparts, and are somewhat dorsoventrally flattened. The legs in the first segment are modified to form forcipules, poison claws, that deliver poison to prey like spiders and cockroaches. As these animals are all predatory, millipedes bear 2 pairs of legs per diplosegment, a feature that results from embryonic fusion of adjacent pairs of body segments. Are usually rounder in cross-section, and are herbivores or detritivores. Millipedes have visibly more numbers of legs as compared to centipedes, although they do not bear 1,000 legs. Figure 9. Subphylum Crustacea. Crustaceans are the most dominant aquatic arthropods, since the total number of marine crustacean species stands at 67,000. But there are also freshwater and terrestrial crustacean species. Krill, shrimp, lobsters, crabs, and crayfish are examples of crustaceans. Figure 9. Terrestrial species like the woodlice, Armadillidium, spp. Also called pillbugs, roly-polies, potato bugs, or isopods, are also crustaceans, although the number of non-aquatic species in this subphylum is relatively low. Crustaceans possess 2 pairs of antennae, mandibles as mouthparts, and, biramus, 2 branched appendages, which means that their legs are formed in 2 parts, as distinct from the uniramus, 1 branched myriapods and hexapods. Figure 10. Unlike that of the hexapoda, the head and thorax of most crustaceans is fused to form a cephalothorax. Figure 11. Which is covered by a plate called the carapace, thus producing a body structure of 2 tagma. Crustaceans have a chitinous exoskeleton that is shed by molting whenever the animal increases in size. The exoskeletons of many species are also infused with calcium carbonate, which makes them even stronger than in other arthropods. Crustaceans have an open circulatory system where blood is pumped into the hemocoel by the dorsally located heart. Hemocyanin and hemoglobin are the respiratory pigments present in these animals. Most crustaceans are dioecious, which means that the sexes are separate. Some species like barnacles may be hermaphrodites. Serial hermaphroditism, where the gonad can switch from producing sperm to ova, may also be seen in some species. Fertilized eggs may be held within the female of the species or may be released in the water. Terrestrial crustaceans seek out damp spaces in their habitats to lay eggs. Larval stages, nauplius, and zoea, are seen in the early development of crustaceans. A cypress larva is also seen in the early development of barnacles. Figure 12. Crustaceans possess a tripartite brain and 2 compound eyes. Most crustaceans are carnivorous, but herbivorous and detritivorous species are also known. Crustaceans may also be cannibalistic when extremely high populations of these organisms are present. Subphylum Chelicerata. This subphylum includes animals such as spiders, scorpions, horseshoe crabs, and sea spiders. This subphylum is predominantly terrestrial, although some marine species also exist. An estimated 77,000 species are included in subphylum Chelicerata. Chelicerates are found in almost all habitats. The body of chelicerates may be divided into 2 parts, prosoma and opisthosoma, which are basically the equivalents of cephalothorax, usually smaller, an abdomen, usually larger. A head tagmum is not usually discernible. The phylum derives its name from the first pair of appendages, the chelicerae. Figure 13. Which are specialized, claw-like or fang-like mouthparts. These animals do not possess antennae. The second pair of appendages is known as pedipalps. In some species, like sea spiders, an additional pair of appendages, called ovigers, is present between the chelicerae and pedipalps. Chelicerae are mostly used for feeding, but in spiders, these are often modified into fangs that inject venom into their prey before feeding. Figure 14. Members of this subphylum have an open circulatory system with a heart that pumps blood into the hemocoel. Aquatic species have gills, whereas terrestrial species have either trachea or book lungs for gaseous exchange. Most chelicerates ingest food using a preoral cavity formed by the chelicerae and pedipalps. Some chelicerates may secrete digestive enzymes to pre-digest food before ingesting it. Parasitic chelicerates like ticks and mites have evolved blood-sucking apparatuses. The nervous system in chelicerates consists of a brain and two ventral nerve cords. These animals use external fertilization as well as internal fertilization strategies for reproduction. Depending upon the species and its habitat, parental care for the young ranges from absolutely none to relatively prolonged care. Section summary. Nematodes are pseudocoelomate animals akin to flatworms, yet display more advanced neuronal development, a complete digestive system, and a body cavity. This phylum includes free-living as well as parasitic organisms like Caenorhabditis elegans and Ascaris, spp. respectively. They include dioeceous as well as hermaphroditic species. Nematodes also possess an excretory system that is not quite well developed. Embryonic development is external and proceeds via 3 larval stages. A peculiar feature of nematodes is the secretion of a collagenous, chitinous cuticle outside the body. Arthropods represent the most successful phylum of animal on Earth, in terms of the number of species as well as the number of individuals. These animals are characterized by a segmented body as well as the presence of jointed appendages. In the basic body plan, a pair of appendages is present per body segment. Within the phylum, traditional classification is based on mouthparts, number of appendages, and modifications of appendages present. Arthropods bear a chitinous exoskeleton. Gills, trachea, and book lungs facilitate respiration. Sexual dimorphism is seen in this phylum. An embryonic development includes multiple larval stages. Thank you for watching BookTube channel. Make sure to smash the like and subscribe buttons to receive notifications about new contents.